I'll introduce some background to the innovation stream, and then we'll head into the second set of flash talks.
Great. Thanks, Megan. Thanks, everyone. I'm delighted to be able to talk to you guys today to outline the exciting innovations in this stream. So it has a very broad aim, of course, is to translate the use of especially new immunological and diagnostic platforms, but also new practice approaches to address what are current clinical challenges, especially in the care of patients with cancer. So here are just a few areas that I wanted to highlight. For example, the pioneering the use of immune profiling for infection risk prediction to address the fact that now clinical assessment of infection risk is becoming more and more complex as patients get more and more courses of treatment. There's also interest around in-depth assessment of immunity, response to vaccination, microbiome changes, especially in a setting of new generation therapies, and trying to understand that a little bit more. There's also, of course, new generation molecular diagnostics to address our current limitations of diagnostic assays, especially significant delays to identification of pathogens of interest. And of course, putting it all together within the stream, we're interested in practice changing and certainly practice defining clinical trials. So the hope is to produce outcomes from new generation of new knowledge that influences practice, but also validation of new technology and platforms, especially from the research setting, where we can then transform clinical practice in several areas. So next slide, please. So just for the next two slides, just to highlight some of the, track record and transformative studies that we have launched. So the first ones are around the importance of 2D studies, establishing as well as refining analysis approaches and pipelines, so that it then makes it a lot easier for the subsequent studies to follow, which a few collaborators will highlight in a few short moments. So just as a typical example for immune profiling, we pioneered the approach of using PMAs together with our WEHI collaborators, the approach of using PMA stimulation to try to look at more functional profiles and looking at it and taking a whole system sort of level approach from cytokine analysis to RNA sequencing to see whether we can predict which patients will develop infections in a 3-month period following sample collection. And I see Lewis online, and he'll outline some of our collaborations in a few more details later. So then also, I think with regards to vaccinations and is for our high-risk hematology patients, especially those B-cell depleted and expected to produce poor antibody responses, is actually studying more than antibody responses from neutralizing antibody assays, but also cellular responses, but again, being quite specific, like epitope-specific cellular responses and looking at the T-cell receptor repertoires. So here are some of the publications that have been produced from these early studies. Next slide, please. And we're of course very pleased that our department and our center has produced several practice-defining clinical trials that have certainly changed practice globally. So ranging from diagnostics, from evaluation of PET versus CT for high-risk patients in the PIPIN study, which demonstrated a higher rate of antimicrobial rationalization with the use of PET, prevention studies that Monica has already highlighted from the interferon alpha nasal spray, which was able to reduce incidence of COVID-19 by 40% and recently accepted for publication in CID. And then looking at interesting ways of approaching vaccination from heterologous schedules for influenza vaccination, and more recently, whether single versus double dose or whether we use adjuvant vaccines can influence clinical practice, and you can see those findings there. And you can see a common trend that all these trials have been published in top of field journals. So we're very delighted by that and hope to continue this outstanding track record. So I might pass on to the next speaker, who will then outline all the interesting studies that they're doing.
Thanks, Ben.
And I think
took the words out of my mouth. Yep. Next, we've got Lewis Williams from the WEHI presenting on the fantastic collaborations that we've built up with those guys.
Beautiful. Thanks so much. Yep, so for those who I haven't met, my name's Lewis and I work with Marcel at the WeHi. And I'm just going to quickly run through some examples of the capabilities we have and what we can do for you. So typically we will receive your patient blood samples as PBMCs or plasma. And from here we can do a range of analysis. So for example, we've been doing cytokine profiling using the Luminex. This is a multiplex system where we can quantify up to 80 immune-related cytokines from a small amount of sample. The main advantage of this is the precise quantified readout. In comparison, mass spec gives a much wider scope of view over several thousand proteins, but we now also have the option to run quantifiable mass spec on any biomarkers identified, which can be a huge advantage if it's a poorly understood protein or there's not readily available ELISA kits. And lastly, large-scale whole genome RNA sequencing is a fantastic tool that allows an unbiased overview of the gene activation status in your patient bloods. We can either provide that data analysis as a list of genes or group them into functional pathways for more relevant interpretation. Ultimately, combining the data from these three techniques with clinical presentations allows us to associate the immune signatures relating to any clinical variables such as infection or transplantation complication. And one last collaborative piece we've been working on is CRISPR diagnostics. This is a platform we've been working on for a few years now, namely with GAB. And we can take plasma samples or similar and develop a test for a pathogen of your choice. So taken together, the diagnostics and biomarker discovery we offer can lead to the early and accurate prediction of infection and help direct personalized medicine. And so with these techniques, we've been working on a few projects already, which include Picnic with Gab, CAR-T and Prim with Ben and Gemma, and Crest with Beatrice and Michelle. And it's been quite a successful collaboration over the last few years, resulting in 5 publications and another 3 in various stages of preparation. So looking forward to working with you all. Thank you.
Thanks, Lewis. Next, we're going to hear from Gemma.
Thanks, Megan. So moving on from Lewis's explanation of our translational applications, we have the Implement study undertaken by myself, Associate Professor Bente, in conjunction with our WeHi collaborators in the Cellular Therapies Unit. And this is looking at CAR T-cell therapy in the treatment of lymphoma and having novel and translational ways to understand immune function and infection risk in CAR T-treated patients, because there's a real gap in our current understanding and a failure of clinical models based on tests that we already have to accurately predict infection in these patients. So what we did was prospectively sample patients treated with CAR T therapy at various time points in their journey, and then use these cells, as Lewis has outlined, to undergo RNA-seq and bulk cytokine signaling to look at associations between these immune signals and subsequent infection risks. And we were able to identify some key kind of findings within these exploratory hypotheses that will spur on future work, including cytokines that were associated with both early and late infections, as well as a dampening of immune signals between the time of CAR T infusion and later time points. Taken together, it gives us a much more nuanced understanding of how someone's immune system is responding post-CAR T, and we hope to carry this work forward with future projects in broader cohorts together with our collaborators at WeHi. Thanks, Megan.
Thank you, Gemma. Next, we have Michelle Yong presenting on behalf of Beatrice Sim, who's currently overseas.
Correct. Yeah, no, thank you. And following on from Lewis's introduction, so Beatrice has been working on looking at CMV and sort of viral immune biomarkers together with WeHi. So we've discussed that CMV is a common infection following transplantation, and we collected a prospective study of almost 120 allo stem cell transplant patients looking at novel immune markers, including the quantiferon CMV, the quantiferon monitor, and more recently transcriptomic analyses to better personalise and stratify CMV ongoing risk, which remains the most frequent opportunistic infection post-allo transplant. We made some findings that CMV-specific immunity fluctuates in the post-transplant period, But we did find that with the response correlates very well with the quantiferon response beforehand, so that a reactive result led to a lower peak viral load in the subsequent 6-week period. The other major finding was that the individual T cell mitogen response as part of the quantiferon correlated very well with clinically significant CMV infection. There some data and PBMCs have been used to look at much more detailed transcriptomic profiling. And as you can see here, that we've identified a number of markers which are upregulated and downregulated in the context of clinically significant CMV infection. So taking this on board and expanding some of this work, particularly using the mitogen into a national renal transplant cohort, Beatrice is aiming to look at patient-specific immune profiles to better determine risk and resolution of infections, with the plan of using these immune markers to improve care in the future. Thank you.
That's exciting. Thanks, Michelle. Next, we have Ben, back to Ben Tay.
Hi, everyone. Pleased to sort of talk about IMPRINT, which is very much a new study that we've just launched. So I think the issue is, as I've outlined a little bit earlier, that the impact, especially of new generation therapies for hematological malignancy on whether someone retains immunity to a range of vaccine-preventable infections, as well as responses to vaccination, remains quite unknown. So I think that there's certainly gaps there about what happens to the immunity. Will patients even respond to vaccinations while they're receiving these treatments? And then broadly, are there better surrogates of protection beyond antibody responses? As I touched on earlier, B-cell depleted patients are, not surprisingly, expected to produce poor B-cell responses, but perhaps cellular responses which are retained would be continue to be useful. So what we have done is launch a prospective cohort study with fixed time point sampling during the treatment, but also following receipt of vaccination into sort of 3 broad cohorts of patients, if you like, with tend to have more long-standing disease, such as myeloma, CLL, non-Hodgkin's lymphoma, for aiming for a total of 120 patients. And the examples of new generation treatments that we're after are like bi- and tri-specific therapy-treated patients, of course, expanding on cellular therapies, looking at drug-antibody conjugates, as well as new generation targeted therapies. So I think the impact, again, would be new knowledge to help define and transform our clinical vaccination practice, programs, as well as policies. And certainly the repository of samples that were built up, allows us to then expand into our work around immune profiling as well. Next slide, please, because I think that's all for IMPRINT, but I believe I'm also presenting the next slide. On behalf of Dr. Victoria Hall, who's currently overseas. So her project is around respiratory viral infections in patients with heme malignancies. And we do know that there's certainly high morbidity and mortality from respiratory viral infections in this patient group. And as with, depending on the type of treatment they receive, such as B-cell depletion, we do know that's reduced immunity from prevention strategies, including vaccination. So I think it's important to address the gaps, which is with these new therapies, what's happening with the epidemiology, the burden, as well as the immune response to these respiratory viral infections. And, to think about better ways to prevent infection, whether that's smarter ways to vaccinate our patients, or perhaps the role of long-acting monoclonal antibodies to prevent infections. And she's done an amazing job looking at certainly 4 different studies, ranging from longitudinal cohort studies like Prosecco for COVID and Rustic, which is currently ongoing. And thanks to the fantastic contribution and support from the clinical trials team, as well as the laboratory team, looking at the epidemiology of respiratory viruses across at least 6 centers around Australia, and with a new collaboration with Singapore as well. There's also, she's completed a immune profiling cohort study looking at the use of long-acting monoclonal antibody as a prevention, which is tixagevimab, sulgevimab, even though it's no longer in current use, the extrapolation of what to do with future long-acting monoclonals is very important. And of course, a pivotal randomized clinical trial of influenza vaccination strategies, which was published in NEJM earlier this year. So the major findings are certainly, as summarized below, the outcomes of COVID-19. We were able to demonstrate in this large national multi-center study that the outcomes have improved over time, and there was support for early treatment. There's a proof of principle study for using pre-exposure prophylaxis long-acting monoclonals, and then as outlined as well, no sort of benefit of using adjuvanted or extra doses of vaccines for our patient cohort, so practice reinforcing, which is great. And I might pass it on to the next speaker.
Thanks, Ben. Next is, hopefully we've got Liv Smybert online, but Monica's on standby. Yep, go take it away, Liv.
Hello, everyone. Today I'm going to be chatting about, I guess, the part of the stream that is looking at the gut microbiome. And so we know increasingly that our gut bacteria are essential for maintaining our state of health and wellness. And when our gut bacteria get out of balance, that can lead to poor health outcomes. And that's certainly no exception in the setting of immune compromise. So the purpose of my PhD work, which is primarily being supervised by Monica at the NCIC with collaborations with the Royal Melbourne and the Austin Hospital, has been to try and explore the role that the microbiome plays in the setting of a variety of immune compromised cohorts. So we've been collecting stool samples before, during and after immune compromise in a group of allogeneic stem cell transplant patients, liver transplant patients and recipients of CAR T to try and see if we can identify predictors or biomarkers that may herald subsequent complications, including infection, which then may hopefully direct us to novel opportunities to intervene and manipulate the microbiome to improve outcomes for our patients, and whether that is subsequent studies that may involve fecal microbial transplant, consortium microbiological products, or simple things such as dietary interventions is sort of the objective of this sort of preliminary PhD work. And so I think in the interest of time, I might just leave it there.
Short and sweet. Thanks, Liv. I hope we get a little cameo. If you stay on, leave your camera on, and we'll have a little camera. Yeah, great.
Yep.
Thank you. Next, we have Michelle Yong.
Thank you. So I'm presenting the Febrile project, which is very aptly named, a prospective study looking at fever and novel sort of biomarkers and translatable sort of novel assays in the immune compromised host. So no doubt the greatest clinical problem that we undertake as clinicians is undifferentiated fever. And not only is the spectrum of pathogens wide and common in this group, it got anywhere from fungal to viral to bacterial and multi-resistant bacterial, but also the differential is wide between fevers relating to a pathogen and fevers relating to the underlying chemotherapy or immunotherapy or cytokine release syndrome. So there's a big gap there, and so the methods of this study is that we will prospectively collect in a multi-centre study 247 patients with suspected fever, collecting blood at baseline and over the first week, and looking at sort of a 30 and 90-day mortality and outcomes. I've collaborated with a number of industry partners to look at these novel assays, which, although some of them have been evaluated in a sepsis cohort, none of them have been conclusively validated in an immune compromised host. So some of these assays involve very rapid PCR-based pathogen detection, which is multiplex and which includes a number of fungus and yeast of interest. And some of these assays are also provide a sort of predictive scoring with detection of upregulation and downregulation of different sort of cytokines, which can help differentiate between high sort of a high septicite sort of score or sepsis, or a low score, which would then allow us to safely manage the patient in the ambulatory setting. We also have some other techniques, and the aim is to generate new knowledge which will help risk stratify our patients who more and more want ambulatory care at home versus the hospital. And we hope that this will generate lots of new knowledge for our patients to come. Thank you.
Thanks, Michelle. Next, we have Emily Klemeski from the lab.
Thanks, Megan. Today I'll be speaking a bit about the ECLIPSE study led by Dr. Zoe Nero. Invasive fungal infections caused by Lamentospora prolificans and Scedosporium species are becoming increasingly prevalent in Australia. They most commonly occur in immunosuppressed patients with hematological malignancy or HSCT and are often life-threatening with a 75 to 100% mortality rate for L. prolificans infections. The most common method of diagnosis is fungal culture and microscopy, which is extremely time-consuming, taking up to 7 days for a diagnosis. Currently, patients are being treated with liposomal amphotericin B, administered as an antifungal treatment based on symptoms suggestive of IFI without an actual microbiological diagnosis of a fungal species. The median time of death to post-IFI diagnosis is 6 days, indicating that there is a growing necessity for early detection methods with high sensitivity and high specificity. PCR is a quick detection method that can be done in only a few hours, although there are currently no PCR-based assays targeting L. prolificans and Scedosporium species. The ECLIPSE study will focus on developing and validating a targeted PCR assay for L. prolificans and Scedosporium species, as well as determining the sensitivity, specificity, PPV, and NPV of the assay. The NCIC lab is working in collaboration with Westmead Hospital and the Peter Doherty Institute to develop an in-house PCR assay to be able to accurately diagnose 4 Lopra and Scedo species. The impact of this study will be to accelerate diagnosis time for invasive fungal infections caused by L. prolificans and Scedosporium species. And I think I'm done. Thanks.
Thanks, Emily. Next, we have Abby.
Thanks, Megan. So I'm talking about some PET studies that are underway. Essentially, as a nice segue from our last presenter, basically diagnosing invasive fungal infections is very challenging and time-consuming and often invasive, with bronchoscopy and biopsy usually our approach to diagnosing infection. So in collaboration with the nuclear medicine team and Professor Michael Hoffman at Peter Mac, we are undertaking this specific study, which is a first-in-human study pilot of 10 patients who have diagnosed, proven or probable invasive aspergillosis, and we will be undertaking a Siderophore radiolabeled PET scan, which is essentially, the idea is that TAF-C is a Siderophore that Aspergillus produces, which then scavenges iron and brings it back to the Aspergillus, and it helps it to reproduce. Now, we can produce TAF-C in the lab and radiolabel it with gallium, which is where the iron would normally bind in that molecule. So essentially, the idea is that the Aspergillus will take up the TAF-C and then be radiolabeled with gallium. So essentially, then we can image the patient with PET-CT, and the idea is to detect the Aspergillus, being a pathogen-specific scan. And so essentially, we image at 15 minutes, 60 minutes, and an optional 3-hour capture. And the image below on your left is essentially the first patient that we recruited to the study, and at 15 minutes we could see that there's quite avid uptake in his lungs at the sites where we have known Aspergillus infection. So that was a nice proof of concept, and it appeared safe. And so we've now recruited 4 patients out of 10, and we're very keen to continue recruitment and hopefully take this forward into a more clinical space. Secondly, another study that's just up and running now is called the PET-IMI study, which is basically a multi-centre with Austin, Royal Melbourne and Peter Mac retrospective study looking at the sensitivity of FDG PET-CT for invasive mould infection as well as microbacterial infection, because we have case series showing that PETs are quite sensitive, are able to detect infection, but we don't know how truly sensitive they are. Can we rely on it? So this will try and answer that question in a multi-centre study. So we look forward to the results of that. Thank you.
Thanks, Abby. Stay online. I think you're up next anyway. But just want to say thank you to the innovation stream. That's just, again, an extraordinary breadth and depth of projects and lots of really exciting work. So I'll hand back to Abby, stream manager for implementation, to give us a bit of background, and then we'll be into the home stretch with our final 2-minute flash talks.
Thanks, Megan. So essentially, the implementation stream, which is led by Professor Karen Thursky in collaboration with the Health Services Research and many other partners in the precinct, aims to use implementation science to inform and scale optimised care pathways for things that are important in patients with cancer, including sepsis, low-risk neutropenic fever programs, vaccination programs, antimicrobial allergy, championing nurse practitioner models of care and antimicrobial stewardship, and in particularly to harness the EMR or electronic medical record platforms and digital technologies and harnessing cancer care networks to optimise care delivery, including virtually and enabling access across the state and nationally. Next slide, thanks.
All right. Yep.
So just briefly, and sorry, this is a little bit small, but essentially it's trying to highlight the breadth of this work. So we bravely undertook the production of the Australasian Neutropenic Fever Consensus Guidelines, which was a big collaboration across Australia and New Zealand, looking at expanding the coverage of the neutropenic fever guidelines to all aspects of neutropenic fever prevention and management, with 8 chapters, including the principles and practice of approach, antibacterial prophylaxis, AMS and IPC, initial management, subsequent management, ambulatory care management, as well as at the bottom there, the patient and carer education, which is a new piece. And it was a really massive effort, nearly at the end now, 53 authors across medicine, pharmacy, nursing, nurse practitioners, microbiology and consumers, now including paediatrics as well as adult, which is a new addition. Across New Zealand as well as Australia is also a new addition, and endorsed by 10 stakeholder organisations. Next slide, thanks. A couple of examples of implementation impact and work led by Ben Tay, looking at consumer co-design in vaccination models in conjunction with Maya Krishnasamy, who is pictured here, looking at the barriers and enablers to uptake of vaccination, including novel vaccination approaches in Australia. And this was qualitative work looking really with consumers to deeply understand what is a barrier, but also what are the enablers to improving vaccination uptake and study in vaccination in general. Also, implementation work into how to better embed antibiotic allergy assessment and practice into electronic health records. Basically, we were able to implement self-assessment questionnaires, best practice advisories to highlight those who have antibiotic allergy and automatically enact a referral. And through the Safer Care Victoria collaboration, we were able to show improved antibiotic allergy assessment and management plans, which greatly improved patient care. Next slide, thanks. So this is a big picture space and lots of big works in progress in collaboration with multiple partners. So some of the examples that are still currently underway, including looking at sepsis and the EMR, which Belinda Lambros will highlight, looking at early recognition and better capture of an identification of sepsis and streamlining management. We'll hear from Michelle Yong about low-risk neutropenic fever and ambulatory care, reimagining that in the new era, looking at models of infection detection and management. A big piece looking at an EMR embedded trial with early cessation of antibiotics in neutropenic fever. This is off the back of Gab Heusler's work in paediatrics we'll hear more about, but Nikhil Singh will tell us more about this project, including implementation sub-studies in collaboration with Lisa Guccione, pictured on the right, surveying patients and clinicians about how their assessments of the intervention itself of early cessation, but also the EMR embedded trial platform, as well as Gang Chen pictured there, who we're working with from a health economic analysis perspective. And also our researchers looking at getting a better scope of low risk neutropenic fever epidemiology as well as high risk. Next slide, thanks. That's it.
Great, thanks Abby for that introduction. So our next slide is pre-recorded. I won't get it caught out this time. Just give me a thumbs up, make sure you can hear it. We'll be hearing from Belinda Lambros, who is our nurse practitioner.
Sepsis is a life-threatening medical emergency caused by a dysregulated response to infection, and it can cause patients to deteriorate rapidly. Cancer patients are particularly vulnerable
being 10 times more likely than the general population to develop sepsis within the first year of their diagnosis. Clinicians today face a challenging environment, a post-pandemic surge in complex cases, alongside the rapid rise of digital tools like artificial intelligence and electronic medical records. This project applies the Learning Healthcare Systems framework to explore and optimise the sepsis care pathway within this evolving landscape. Using a method called model transparency, we created a clinical workflow model and digital phenotype to understand the sepsis workflow in detail. Clinicians must be able to understand the digital tools they are using and not just rely on blind trust of the IT builders, just in the same way that patients need the clarity of the informed consent process. The project had 3 aims. Firstly, to validate the current sepsis workflow and alerts built in Epic, to optimise those workflows, and finally, to validate reporting on compliance measures to ensure accurate data was available on patient outcomes. The first major step was business process mapping, comparing bedside practices and EMR workflows with expectations from the end users, including bedside clinicians and the sepsis team. Real-world retrospective data will be analysed to find discrepancies and areas for improvement. A large dataset will also allow for future research and further optimisation of the workflow. Ultimately, this project has produced a sepsis care path digital phenotype, harmonising workflow roles and aiming to improve sepsis recognition and compliance. It offers a reproducible model for other healthcare services using Epic, as well as a blueprint for validating clinical pathways within the digital landscape.
Thank you, Belinda's recording. Next, we have our second nurse PhD, Alison Lemo.
Thank you. Hi, I'm a PhD candidate at Peter Mac and NCIC. The problem I'm looking at is, as we know, sepsis causes approximately 20% of global deaths each year, and delays in the early recognition and management of sepsis greatly increase patient morbidity and mortality. So the gap that I've identified with my supervisors is nurses are the largest group of healthcare providers, but we don't know what barriers and enablers they encounter in the early recognition and management of sepsis within high and low resourced Australian healthcare settings. So the methods I've employed, firstly, I've done a scoping review of nurses' contribution to the acute sepsis care. The findings from this scoping review so far are that there is a role for the specialist experienced nurses in lower resource settings, and it raises questions about the risks versus benefits of having an expanded scope for nurses in some settings to include perhaps interventions such as restricted IV fluids and first dose antimicrobials. But it also raises questions about postgraduate training requirements for these nurses, and there was a lack of consistent nomenclature with specialist roles. The second study I'm doing is a qualitative interview study exploring barriers and enablers to nurse-initiated sepsis care across metro, regional and rural and remote Australia. And I got 15 nurses who were interviewed, and the preliminary themes that are emerging from that, again, are around challenges remote and regional area nurses face working without a doctor on site, caring for the acutely deteriorating patient, and also issues relating to Indigenous care, healthcare outcomes and sepsis, and also improving university-level sepsis education for nurses who are enrolled in registered nursing studies. The last study will be a national CAP survey, knowledge and attitudes and practice survey of these barriers and enablers to quantify findings. And the impact of this study, of this PhD, will be the development of targeted evidence-based implementation strategies that promote timely sepsis recognition and management by nurses across high and low resourced Australian healthcare settings. Thank you.
Perfectly to time. Thanks, Alison. Next, we're going back to Michelle.
Yeah, so I'm going to talk about the ADAPT study, which is a large MRFF-supported study looking at trying to achieve durable AML responses in patients treated with venetoclax azacitidine. So I'm really excited because this is one of our first sort of embedded collaborative RCTs with the Australasian Lymphoma Leukemia Group, the ALLG, which is the premier sort of society in our region. The problem is that febrile neutropenia management is not standardised in AML on Ven-Aza, and we need to keep up to date with the current treatments, with Ven-Aza being used in patients who are otherwise have comorbidities, who are older, and sometimes often with a palliative intent. So the ability to deliver safe care and fever care at home whilst on neutropenic is really paramount. So this will be a randomised controlled trial of ambulatory care, implementing our sort of ambulatory care model versus standard of care, which is to remain inpatient whilst awaiting neutrophil recovery. So we will look at instituting a nationally oral antibiotics and supportive care for those patients who are deemed eligible to be randomised, with the endpoint being 4 weeks of fever-related hospitalisation. And for this work, it introduces one of our upcoming PhD candidates, Dr. Jessica O'Keefe, who will not only help support the national running of this RCT, but also look at specifically infection-related mortality in AML and assess consumer and clinician-related research priorities in this new field of AML treatment. Thank you.
Thanks, Michelle. Next, we're going to hear from Morgan Rose.
Afternoon all. So antibiotic allergy is a huge problem in immunocompromised hosts and comes with a number of negative health impacts. And this study that we recently took part in really highlights our role at NCIC as real leaders in antibiotic allergy for immunocompromised hosts. And so this was the prospector study. The main problem is the vast majority of these antibiotic allergies are delayed, which means they didn't happen immediately after starting the antibiotic. And what is unclear internationally is whether one tablet, so if I then go and challenge a patient with that antibiotic again, is one tablet enough to actually prove they're not allergic, even if they tolerate it? So this study was doing two things. One was starting to answer that question, but also was really demonstrating that we could undertake a double-blind placebo-controlled trial in drug allergy. So that's what we did. Once people passed their challenge, they'd either have 5 days of amoxicillin or 5 days of placebo, and no one knew what it was except for the pharmacists, and then assessing to see if anybody had delayed reactions, and was that more common in the amoxicillin group. Pleasingly, what this showed is that yes, we could facilitate this type of study in drug allergy, and has then led to firstly the publication of the results that's just been accepted into NEJM evidence, and then really the impact is that it's informed the subsequent efficacy study Prospector 2, which is international, multi-centre, and has just started recruitment at PMC at Peter Mac as well, and really looking to globally change antibiotic allergy practice for this majority of patients with delayed allergies.
Thank you, Morgan. Next, Violet Zhu, one of our PhD pharmacists.
Thanks, Megan. So I'll be providing a very brief overview of my PhD, which is titled Defining the Framework of Antiviral Management in Transplantation, which is the next frontier of antimicrobial stewardship. So as some of the other presenters have stated, viral infections in transplant are common, and CMV infections are of particular interest in the post-transplant period due to their association with significant morbidity and mortality. The antivirals that we use to treat these infections are toxic, and they're high cost and can lead to rapid resistance when we don't utilize them correctly. So there's really this unmet need to optimize the therapies in this cohort. So the major projects as part of my PhD is firstly a systematic scoping review to elucidate the definition of antiviral intolerance within the literature. The ASCENT study, which is one of the major projects, which will be evaluating the first of its kind antiviral stewardship CMV multidisciplinary team meeting in the allogeneic stem cell transplant cohort, which currently occurs at the Parkville precinct. Third study was around therapeutic drug monitoring for valganciclovir and ganciclovir, and how population PK modeling for this cohort can inform dosing, as well as defining the relationship between plasma concentrations and clinical outcomes, as well as toxicities. We'll also be utilising hospital NAPS, which is an existing national program to assess where the gaps are in antiviral prescribing are on a broader scale. And finally, all of these projects will help inform and develop key metrics for antiviral stewardship programs in Australia, which will be elucidated through a modified Delphi process and broaden the implementation of a lot of these key programs that we've described. So the hope is that the results from all of these projects will help optimize clinical management and improve health resource utilization in the transplant recipients with CMV infections. Thank you.
Thanks, Violet. Next, we have Karen Abanchik, another one of our pharmacist PhDs.
Thanks, Megan. So I'll be talking about the popular study which involved looking at posaconazole plasma concentrations exclusively in the tablet formulation with patients with hematological malignancy receiving prophylaxis. So we know from what Monica outlined earlier in the showcase that IFIs are costly in this group of patients, not only in terms of healthcare costs, but also in clinical outcomes like morbidity and mortality. So the improvements to antifungal prophylaxis are vital. The gaps in this area include limited prospective posaconazole level data with the tablet formulation, as well as the impact of diarrhea on plasma levels across the prophylactic course. And with the improvements of bioavailability with the posaconazole tablet formulation, it's now a bit unclear if routine therapeutic drug monitoring is actually needed in prophylaxis. So we conducted a multi-centre prospective cohort study collecting a rich data set of posaconazole plasma concentrations in this setting to help describe exposure throughout the course of prophylaxis, as well as the covariance influencing therapeutic level attainment. We're using the data to build a population pharmacokinetic model specifically to predict when subtherapeutic levels are likely to be reached in the setting of diarrhea with the oral tablets. And we also want to use the data to explore the impact of routine TDM on the timing and extent of switch to IV posaconazole, as well as on dosing. So the impact of this work will be to understand ways to optimise posaconazole prophylaxis in this high risk group of patients. Thank you.
Thanks, Karen. Next, we hear from yet another PhD pharmacist, Nikhil Singh.
Thanks, Megan. So I'll be giving a brief overview of the ELSA adult trial. So we know that adult hematology patients who receive intensive chemo are often started on antibiotics at the onset of neutropenic or pre-neutropenic fever. And in many cases, the fever is not caused by bacterial infection, yet our standard approach is to continue antibiotics until blood counts recover, resulting in unnecessary antibiotic use. The gap here is that early antibiotic cessation strategies is an understudied area in this high-risk cohort with need of further high-quality data. So what we'll be doing is scaling a pediatric EMR-embedded RCT, as Abby mentioned, to the adult setting across both Peter Mac and Royal Melbourne. We'll be randomising patients to an early stopping arm once they afebrile and meet clinical stability criteria, or to a standard of care arm. So what we'll look at is antibiotic-free days, clinical outcomes, and microbiological outcomes. In addition, we are completing 2 sub-studies, one looking at gastrointestinal microbiome endpoints, another looking at health economics and feasibility of our intervention. So with regards to impact, this is an EMR embedded trial. This is a novel study design that can inform future trial designs, particularly those that are looking to scale existing EMR embedded trials. If we find that early cessation is proven safe, this study will likely change practice standards nationally and impact future guidelines, both either nationally or internationally. Thank you.
Thanks, Nikhil. Last but not least, I think, yes, we were just wanting to hear from Associate Professor Gab Heusler, who is a NCIC alum and superstar. She's now the head of infectious diseases down at the kids down the road. And we're really excited to and pleased to hear from Gab about all of our fantastic collaborations.
Thank you. Thanks, Megan. So I'm here to talk about the paediatric arm of the NCIC, which we've brought under the one umbrella and called this piece program of research Protect. So many of the projects that we've already heard about today, we've been fortunate to be able to incorporate paediatric arms into those studies, and there's a few that I wanted to particularly highlight today. We have all the same issues in paediatric cancer and transplant infectious diseases as we have in adults, but well, fortunately, a much smaller pool of patients to work with, both in size and number. And so the importance is really to ensure that we are very collaborative with our work across Australia to get the numbers we need for the various studies. So the program of research really kicked off with the Australian Picnic study, which was a paediatric febrile neutropenia epidemiology study, which resulted in implementing low-risk febrile neutropenia programs across all 8 sites in Australia and subsequently 14 sites in the UK. That has informed the febrile neutropenia antibiotic duration study, or the ELSA study, that is fully embedded into the EPIC EMR and really served as a platform for building out the adult ELSA study. And that's been led by Colin Butters as part of his PhD. I've also had the pleasure of co-supervising Dr. Hannah Walker, who's a paediatric oncologist, and her study, BREATHE, is looking at the inflammatory profiles of patients that undergo transplant and have pulmonary complications to see whether there's any predictors of these pulmonary complications, and also looking at novel diagnostics in this space. That database has been able to be leveraged for further study looking at CMV infection in this vulnerable population, and Dr. Miles Horton is a postdoc working with our team looking at CMV-related immunity in these patients. And finally, but certainly not least, Dan Yeo, a postdoc student now, completed his PhD, has really spearheaded the paediatric invasive fungal infection work across the country, and his study, Pacific, has really pulled together important epidemiological information about risks of fungal infection in this population, and he's also built out some novel diagnostic work within that piece of work also. Thank you.
Wonderful, thanks, Gab. That brings us to the conclusion of today, and I'll hand over to Monica to close. But firstly, just thank you to everyone involved in putting all of this together. It was a huge piece of work, and it's just been fantastic to listen to all your talks today. Thank you.
Thanks, everyone, and thanks so much for giving so much information in such a short space of time. It's amazing, but I think we've come in right on time. So thank you to all the presenters who've presented their really outstanding work and, you know, world leading, really pushing the envelope forwards for improving outcomes in cancer patients. So I'm really hugely proud of the whole group, of our collaborative endeavours, and also the research that's being done. And I guess just a final word of thank you to all of our collaborators, which we wouldn't be in the position that we're in today. So some of them are listed here, but I'd also like to single out NCAS, the National Centre for Antimicrobial Stewardship, for all the work and collaborations in Einstein and the fungal surveillance, and many of our projects have both antimicrobial stewardship and a patient focused lens. So I'd also like to thank RMIT for their work on Einstein, and we've got a very good collaboration with them. We've heard about our work with the Walter and Eliza Hall Institute for the immune profiling. And again, the Centre for Antibiotic Allergy and Research at the Austin is a very long-standing and strong collaborator with us on the antibiotic allergy work. And of course, we also collaborate now even more strongly with the Royal Children's and the Murdoch, with Gab's new role leading infectious diseases there. So I think we're very pleased that people were able to join us from around Australia and from our collaborators, and in Singapore as well. And we really would be looking forward to growing these collaborations over the next 5 years going forwards. I also want to thank Megan, of course, who was the Sergeant Major coordinating all of this. Without her precise brain and logistical brain, this wouldn't have happened. And so thank you very much, Megan, for a huge amount of work in getting all of this together, getting everyone whether they liked it or not, to submit their slides on time, and getting all the slides together for this, and also the people who've helped on site in the various rooms where we've been running the talks. So again, big thank you to everyone. I think the future is very bright for our group. And I'd just like to say to the amazing researchers again, thank you for sharing your work with us today. And to our collaborators, we look forward to ongoing collaborations and expanding collaborations. Thank you. I'll close there. So bye, everyone.